It's my pleasure to introduce our first speaker, who is Jodie Malcolm from the University of York. She's a postdoctoral research assistant there, specializing in advanced microscopy and cancer phenomics, where her work explores the dynamic cellular changes that underpin cancer progression. Additional to that is that Jodie is deeply committed to improving global access to advanced microscopy and bioimage analysis tools, particularly for researchers in resource-constrained settings. So it's my great pleasure to welcome Jodie to the podium.
So, hello everybody. It is a real pleasure to be here today, and I'm very excited that I have an opportunity to share our story and the work that myself and this fantastic board of people behind me have been doing over the last year or so to try and improve global access to advanced microscopy and the image analysis tools. So very briefly talk about our research at York, and then hopefully that will be a nice insight into how it feeds into our bigger motivation. So in the Brackenbury and Peter O'Toole's lab at the University of York, we're very interested in studying cellular phenomics, so the dynamic changes that occur in cells over a period of time, with a particular focus on breast cancer. And we're very fortunate because we have access to high-end label-free imaging systems such as the LiveSight and the Tomacute. So we have these instruments that allow us to capture these beautiful videos. But having a video is very nice, but we need to actually have a way of measuring and extracting meaningful information from the data that we are presented with. So there's a lot more that meets the eye: how our cells move around the surface, how they interact with each other, how quickly they divide, and their size, shape, and texture can be very informative about the biological processes that are occurring in these cells. It might even help us identify cells that are going to respond to chemotherapy or be resistant. With the advent of machine learning, segmentation, tracking, and single-cell feature extraction has become quite customary now in bioimage analysis. And this is a very shameless plug, but another thing that we've been doing at the University of York is developing a selfie toolkit, which enables us to do this segmentation, tracking, and single-cell feature extraction in one go. And if you are not a software developer or bioinformatician, this has got a graphical user interface that allows you to access these tools open source and with ease. But for researchers in resource-constrained settings, the picture is slightly different. So we've had a very important workshop held at the University of York in May, where we had really important discussions surrounding the key challenges affecting access and uptake of advanced microscopy in these regions. They centre around limited access to education and training, proper infrastructure, not just in terms of like a facility that holds the microscopes, but also having the ability to store and process the large data sets that come with advanced microscopy. Obviously, this follows with economic constraints and then a lack of expertise. And together, this all limits the ability for researchers to generate this preliminary data. And so I hope you can appreciate that these challenges are not standalone, but very much feed into each other and perpetuate each other. So that's really our motivation, is to sort of overcome these challenges and in fact turn them into opportunities so we can see economic growth and enable researchers to generate the preliminary data necessary to pioneer their research in locally relevant health challenges. To overcome this, I'm going to talk today about three key things that we've been doing. So firstly, we've been training custom machine learning models that have been specifically designed to improve the segmentation for low-cost microscopes, which I'll talk about shortly. We also wanted to show that our selfie toolkit can be used for a selfie phenotype extraction on these low-cost microscopes. And finally, I want to talk about how we've optimized a very ultra-low-cost 3D printed microscope called the OpenFlecture to enable researchers to generate this preliminary data. And it's not working. Okay. So we're a part of a fantastic community of researchers around the world. So I'm based at the University of York, but we've also got a really fundamental relationship with the Broad Institute, so Beth Simony, also BioRTC and IDM with Karen and Mahmood, and Anna Paula and Leong Chu as well. So we've got a really wonderful hub of researchers who are all connected and sharing the same sort of motivations in research. We also have some industrial partners, so we're working very closely with AstraZeneca and GSK, and the third partner here is Iolite, who is another example of low-cost microscopes, which I will go into more detail about. So in terms of identifying low-cost microscopes that can be used for these phenotype extractions, we've been working with 3 different types of instruments. Hopefully. There we go. So we've got the open Fletcher and iolite, as I've mentioned, and also a hollow monitor, which is a very low-cost, comparatively QPI system. But it's the story of the open Fletcher and the iolite that I will be sharing with you today. So the iolite is about £2,500, so I think that's about 5,000 Canadian dollars. And this is the type of data set that you might be able to generate. So this is time-lapse microscopy for some human memory epithelial cells. The iolite microscope itself is a really versatile instrument and can fit quite nicely and compactly into tissue culture incubators. And with the efforts of a fantastic colleague, Lur, from the Broad Institute, we have been working tirelessly to develop a custom cell pose model. This was a real feat for Lee and Lur, and I have to really commend her for her efforts. It's a real wonderful project to be working on. It's a pleasure to work with her. But now we have this custom cell pose model that's been specifically designed to actually do the segmentation of data sets generated from the iolite, and this is open source and accessible. But for researchers in low-resource settings, we were very excited to invite Anna Paula Lima from UFRJ over to the University of York to do some preliminary studies using our iolite microscopes. So Anna is very interested in studying leishmaniasis, and we conducted a very simple experiment using our low-cost microscopes where primary macrophages were cultured in the presence or absence of leishmania. And this is the data set that we were able to generate. And if you look carefully in the leishmania-infected macrophages, you might actually be able to see some parasites swimming around in the frame as well, which is very exciting. But importantly, to actually get some meaning out of this, we wanted to show that we could actually put this data set into our selfie toolkit, and this is the kind of results that you might expect to get. So selfie enables us to identify important discriminatory features that change over a time series, and between the 2 populations of uninfected and infected macrophages, we were able to see separation and identify the features that are most responsible for this separation between the 2 populations, namely around texture, but there was also some changes in the cell density, cell size, and movement as well. Now moving on to the story of the open Fletcher in my last couple of minutes. So these open Fletchers are 3D printed microscopes, and their STL files are also open source. And for us in the UK, it costs about £300 to be able to buy the full kit, and you can buy it pre-printed. But for live cell imaging and the sort of research grade type of microscope we want to achieve, there were a few issues. So namely, if we want to put these microscopes into a humid incubator, the electronic components are housed inside the microscope, so that's going to obviously cause some issues with electronic corrosion. So we wanted to move the components outside of the incubator to protect them. In order to do live cell imaging, we need to have an appropriate stage so that we can have a tissue culture vessel on the stage rather than slide clips. So that was a very easy fix of just printing a new type of stage. But in order to do more advanced microscopy, you have to use Blockly, which is reliant on internet access, which is not a given for researchers in resource-constrained settings. So we wanted to enable the microscope to be operated completely offline and be able to do more advanced microscopy without an internet connection. And we also did a final project, which was a very excellent thing to work with an undergrad student who worked on this, which was optimizing the printing material required to enhance durability and improve image quality. So here's a 3D printed schematic provided by our mechanical workshop. We've also incorporated a hammock to sort of also overcome the issues we were getting with vibration, but this is all completely 3D printed, and we've got the electronic components outside, which also feeds into a very cheap monitor, so it can all be accessed and operated outside the incubator. This is our simple graphical user interface that we've designed, so this allows complete operation of the microscope and setting up time lapses completely locally without any internet requirement. And in terms of the materials that we were using, we identified out of 4 potential polymer printing materials that ASA was the most stable and durable of these materials. To print the whole microscope, it cost about £3.15, so we're kind of maintaining the low-cost scheme of the project, and you can be printed within 4 hours. This is the type of data that we might expect to achieve. So maybe it doesn't look as good as what you'd get from tychographic imaging systems, but we can actually see in a dose response here how the cells are behaving. So we've got cells responding to docetaxel. We can see that they're failing to divide and will eventually die, and we can watch the apoptosis, whereas the control cells are actually moving and happily dividing around the plate. And as we continue to develop our Open Fletcher pipeline, we have got a segmentation model that is still in progress, but we are able to segment these cells. And from this very early study, we have shown that we can identify a significant separation in the features between the two populations, even with low cell number, using our selfie toolkit. So just to very briefly summarize, I've shown today that we have developed selfie tool previously, and this can extract important biological features from time series data. We have trained the cell pose model on time series data curated with low-cost microscopes, enabling critical questions to be addressed in resource-constrained settings. And we've strategically modified the Open Fletcher microscope for a fit-for-purpose, locally operated, network-independent and incubator-proof microscope capable of generating preliminary data. So there's a lot of people here that I'd like to thank. Thank you to the Wellcome Trust for funding our excellent project. It's a real pleasure to be a part of it. To the BACR for funding me coming here today to share our project. To the organisers for inviting me to come and share our story and for organising a fantastic conference. And thank you all for listening.